Welcome to Axcelead web seminar. My name is Kazuyoshi Aso, Head of Global Business Development, Axcelead Drug Discovery Partners. Today, we have many audience from all over the world. Thank you so much for your time and attending this Axcelead Global web seminar. Axcelead Drug Discovery Partners is a Japan-based drug discovery solution provider. We have a wide range drug discovery capability. Among this capability, today we selected the very unique and interesting our capability and services. The topic, the meeting title is Axcelead Target Protein Degradation Drug Discovery with Axcelead's integrated platform, DegLead. Today's speaker is Hiroki Kamitani, who is a director of science, strategy, and planning. He is a great expert of the target protein degradation field. I hope you enjoy this seminar and you learn about details of our capability and services. And we will happy to take your questions during this seminar. If you have any questions, please use a Q&A box. Don't use the chat and raise hand. Let's get started. I hand over to Hiroki. Please start your presentation, Hiroki.
Thank you very much for joining our webinar. My name is Hirotaka Kamitani. I'm the director at Science Strategy and Planning Division, and they are responsible for the development of services for targeted protein degradation, the so-called TPD. Today, I'm going to talk about the overview and the representative strengths of our TPD services that we call the TPD services DegLead. Here's today's agenda. I'll start with the introduction to general features of TPD, and then talk about our TPD platform, DegLead. Firstly, I'd like to talk about the introduction. TPD is believed to be a true paradigm shift in drug discovery, especially small molecule drug discovery. TPD can tackle undruggable targets such as transcription factors, scaffold proteins, helicases, and so on. In addition, TPD can reverse druggable targets such as kinase because a TPD can avoid or mitigate drug resistance, provide target or cell specificity, and in some cases provide stronger efficacy than inhibitors. However, there are big challenges in the TPD drug discovery. For bifunctional degraders such as PROTAC, the optimization process and the structure-activity relationship is complicated and are sometimes confusing. There are many sites to be optimized in this molecule. In addition, the slight modification of the compound often unexpectedly affects the degradation activity. The difficulties tend to consume a lot of time and resource. Furthermore, discovery of compounds with desirable pharmacokinetic profiles remains a representative challenge. It's possible, but challenge. In addition, in vitro in vivo correlation of ADME is very poor, making rational improvement of pharmacokinetic profiles difficult. For molecular glue degraders, rational or effective methods of discovery have not been established. There are several reasons, and I think one of the reasons is insufficient of special or suitable compound library for molecular glue degraders. Furthermore, advanced and diverse assay capabilities are needed. So we have been developing TPD solutions for service TPD solutions to address these challenges in recent years. From here, I'd like to talk about DegLead platform. Firstly, I'd like to talk about overview. This slide shows the overview of DegLead platform. We have integrated drug discovery capabilities for bifunctional degraders and molecular glue degraders from target candidate. We launched the service in 2023. To our delight, the service has been becoming popular in our clients soon, so the track records have been rapidly increasing. We have already obtained some impressive results. From here, I'd like to talk about our solutions for bifunctional degraders. They contain the specialized chemistry, in vitro assay systems, ADME solutions, and the case studies. This is a specialized chemistry, and it is a high-throughput chemistry for bifunctional degraders. This is one of the popular services in our clients. Our multi-step and high-throughput chemistry using a degrader chemical toolbox for bifunctional degraders can accelerate hit, lead, and candidate discovery. We can synthesize 96 compounds per one batch through 2 to 5 multi-step reactions in 1 to 2 months from planning and completion, including purification and concentration. Regarding degraded chemical toolbox, we have over 50 E3 binders consisting of cerebron and BHL, and there are thousands of linkers in stock. This chemical technology and the degraded chemical toolbox enable to rapidly access to over 100,000 chemical space. Furthermore, each batch and the combination can be flexibly modified due to our system is multi-step. In addition, we have a uniqueness about POL binders. We have a lot of compounds for over 500 drug targets from our legacy. So in some cases, we can offer unique values through our original POI binders. This is slides show also about our bifunctional degrader synthesis. This slide shows our advantage of a high throughput chemistry for bifunctional degraders against our general method. The table summarizes our approach and general method. In general method, high throughput synthesis is conducted by using hundreds of E3 binders with linkers through one-step reaction. This method is very simple and easy to do experiments, but the chemical space is very limited, and the flexibility and versatility is very low. In contrast, our system is very high flexible and versatile. And I explained it in the previous slide. Our technology and the chemical toolbox enable to access our big and large chemical space. In conclusion, our chemistry service can accelerate not only hit, but also lead and candidate discovery from cerebron, BHL, or other binders for hijacking proteolytic systems, including client's own binders, due to the features. In addition, multi-step parallel synthesis of bifunctional degraders are very difficult in terms of technique. But we have a strong confidence to succeed in it. In actual, the success rate of synthesis is basically over 90%, and even 100% is not rare. Next is assay systems for TBD. We have diverse and very experienced assay capabilities for TBD. For example, protein degradation activity, ternary complex activity, binary complex affinity, and the degrader profiling, such as cooperativity kinetics. Today, I'll not go into the details, our systems, our assay systems evaluate synthesize the compounds in a high throughput manner and provide accurate structure activity relationship to be a great support of acceleration TPD projects. Next, I'd like to talk about ADME solutions for bifunctional degraders. In a previous study, we succeeded to discover many orally available bifunctional degraders. However, the membrane permeability and aqueous solubility were not detected by standard ADME assay method. Namely, in vitro in vivo correlation is very poor. To solve the problem, we developed modified CAC2 assay systems to enhance the detection of power membrane permeability. In fact, the modified CAC2 assay systems can detect the membrane permeability of most of orally available bifunctional degraders. In addition, we have established a suitable ADME assay package and assay flow as the total ADME solution enabled to improve in vitro in vivo correlation or bioavailability and even pharmacokinetic profiles in high throughput manner. And this is a case study utilizing ADME solutions and the drug design technologies for bifunctional degraders. We succeeded to discover orally available bifunctional degraders with high probability. In the project for target X, we synthesized about 100 compounds from target X binders utilizing our design technologies and ADME assay systems and ADME assay data. Surprisingly, 90% of newly synthesized compounds were orally available, and several compounds showed over 20% PA. And some compounds showed target X degradation. I think this is a very good case. However, there are difficult cases. But our own capabilities enable to improve the possibility of success as possible for oral drug discovery of bifunctional degraders. Regarding bifunctional degraders, this is the final slide. This is a case study about integrated solutions from POA binders for initial bifunctional degrader lead generation. We succeeded to smoothly identify desirable exit vector in solvent accessible site of POA binders through compound synthesis and binding assay. Next, heat generation was achieved using our high-throughput chemistry and using degraded chemical toolbox to yield a moderate active compound. Next, the initial lead generation stage, we synthesized high-throughput synthesis and individual synthesis, and evaluate them in high-throughput manner. As a result, we can succeed a highly active bifunctional degrader with acceptable ADME profiles. That the total period is about 1 year. In this case, we can deliver promising bifunctional degraders in a short time through our proprietary technologies and compound assets and integrated drug discovery capabilities. Finally, I'd like to talk about the solutions for molecular glue degraders. This is an overview of capabilities for molecular glue degraders, compound library, HDS capabilities, or degrader profiling identification platform. Today, I'd like to talk about the compound library, HDS capabilities, and one of the degrader profiling or identification technology. This is our compound library and HDS capabilities for molecular glue degraders. We have high quality pharma-originated 1.2 million compound library and the proprietary molecular glue degrader focus library. Our molecular glue degrader focused library consists of 6,400 compounds, including about 600 compounds from internal HTS campaigns against over 5 ethyl ligases. The rest compounds were selected from multiple in silico analyses, such as similarity, substructure, pharmacophore, or a docking study of ethyl ligases. As for high throughput capabilities, we have various capabilities and experiences. We can conduct high throughput screening using high-bit, TR-FLET, alpha-LISA, and NANOFLET, SMS, and so on. In conclusion, we believe special compound library and HTS capabilities are unable to discover the promising set point for molecular glue degrader drug discovery with high probability. This is our final capability is molecular glue degrader and DegLead today. This is a omics capability about a global proteomics and the proximity labeling. We have diverse omics capability. Among them, global proteomics and proximity labeling is very important technologies in molecular glue degrader. Global proteomics efficiently identify our downregulated proteins, and the proximity labeling efficiently identify proximity-induced proteins of the by the compound. Now, the combination of this technology efficiently identifies the direct off-targets on new substrates or degradation mechanism of molecular glue degraders. In the field of molecular glue degraders, there are often cases that identification of new substrates or degradation mechanism is very difficult. In such cases, our capability is one of the good solutions for such issues. Now, this is the end of my presentation. We offer comprehensive capabilities at every preclinical stage, including GPD drug discovery research. Thank you for listening.